All right, guys. So if you're watching this video, this is for the 6.1 cell size to surface area volume ratio lab 6.1. And so basically what I'm going to do, this is going to be two videos. So this first video, I'm going to actually run through the lab and see what we see with the cubes. In the other video, we're going to talk about how to fill out that information in your lab book following that template on the slide. So with this, basically the whole purpose of this lab is to collect evidence to explain, okay, why can't cells just continually grow larger? We're made up of trillions of cells. They're really, really small. And I read somewhere that, the kidney cell of an elephant is the exact same size as a kidney cell of a mouse. So we don't see larger cells with larger organisms. We see more cells, more cell division. So why is that? Why can't they just get bigger? Okay. And so that's the whole point of this lab, is to try and understand why that happens and why cells can't survive when they get larger. So basically what we have here is we have three different cubes that represent cells. So we have a small cell, 1 centimeter. We have medium, which is 2 centimeters, and then a large one, which is 3 centimeters. Okay. And so we're going to use these to simulate the cells. So part of this lab, you'll see in the analysis, it says, okay, what do the cubes represent? Well, these represent different size cells. Okay. And then what we're going to do is we're going to put them into vinegar. Okay. Now, what's going to happen is vinegar's slightly acidic. Within these cubes, they have a pH indicator, the bromothymol blue, and it'll turn yellow when it comes in contact with the vinegar. Okay, so as we put these cells into vinegar, we'll start to see that vinegar work through the cell, and the cell will start to turn yellow. Okay, now what we want to connect is the cubes represent the cell, the vinegar represents the nutrients, the oxygen, the glucose, the water, all the things that the cell needs. Okay, and we want to understand that all that material has to pass through the cell's membrane to get inside the cell. So that's going to be the first part. That's going to be one of the pieces of evidence we collect, is we're going to look at, okay, how far did the vinegar make it through these cells? So what we did is we got our vinegar, and then we're just going to gently place our cells into our vinegar. And what we should start to see is we're going to start to see a different color start to form around the edges of these, right? We can already see it's kind of lightening up a little bit, but we're going to wait about 15 minutes. We're going to come back and we're going to look at, okay, was that vinegar or the materials that the cell needs, was it able to make it throughout the entire cell? Because if that cell doesn't get the things that it needs, or if that material doesn't make it through the entire cell, the cell's not functioning properly and the cell's going to die. Okay, so we're going to go ahead and pause this. We'll be back in 15 minutes. Okay, so it's been about 15 minutes with the cubes in the vinegar. So we're going to take them out and we're going to look at what we found. So we can see they're starting to look very different, right? Instead of being that nice blue color, they look kind of pale or clear, almost yellow. Okay, so there's a 3 centimeter, 2 centimeter, and finally, get it, 1 centimeter cube. Okay, so we're going to start with that one. So again, remember, what we wanted to see is if the vinegar represents the nutrients that the cell needs, we want to see, did those nutrients make it all the way through the cell? Well, how do we know that? If they've made it through all the way through the cell, the cell should be this yellow color all the way through. So if we cut this bad boy in half and we look, you can see on there, it's all the same color throughout, which tells us that vinegar was able to make it all the way through the smallest cell. Okay, so now when we go to our other two cells, we can already see, if we look closely at this one, there's a small blue portion in there. Okay, and when we cut that open and look, we can tell that it was close, but that little bit of blue is showing that that vinegar didn't make it all the way through the cell. Okay, and so that what that tells us is that cell would not have gotten all the nutrients that it needs. And then finally, our largest cell, we can definitely see some color in there. And when we cut that open, oh, very satisfying. Look at that. Okay, so we see that dark blue. So what this is telling us is our nutrients only made it through this first small layer, right? And then this entire portion of the cell didn't get any of the nutrients that it needed, okay? So that's really the key thing to understanding this is as we increase cell size, that increases the amount of oxygen that the cell needs. That increases the amount of glucose, the amount of proteins it needs to build. All of that demand, that material demand, is increased as the cell gets bigger. Now, that makes it harder to get the materials that it needs into the cell as well. And we're going to talk about that in the next video, why that surface area to volume ratio matters. Okay, so again, though, last thing, big thing, vinegar represents the nutrients that the cell needs. Each block represents different cell sizes, and this is showing us that that cell didn't get everything that it needed. That vinegar wasn't able to make it all the way through this block, showing that the cell wouldn't get the nutrients that it needed throughout. Okay.